Dang it, I just started recording. Hold on one second. All right, sorry about that. So just to remind people that aren't here today, again, the main thing is to use Chrome if you can. And what was one of the other things that I mentioned? Make sure that you look at the table on the muscular system that does look at the different fiber types distributed between the different segments of the population. So I just got to remind myself to record. All right. So speaking of the nervous system, we did speak about the withdrawal reflex, which is one of the most simplest aspects of the nervous system. And we did a comparison, being that this is animal physiology. It's good to sort of compare and contrast. So we did speak about the cockroach startle response, and again, how similar that is, what are some of the components of it that really help define very similar our withdrawal reflex. So knowing what the nervous system is, how it's organized, what its overall function is, then we got into the basic components of it. Just like any system, it's made up of specialized cells. And the main cell of the nervous system, of course, is the neuron. That's the main functional unit that's responsible for the sensory and motor and integrating functions. You should know the anatomy of it, the different parts of the neuron, what are the functions of the different parts. For example, which part receives stimulus, which part releases neurotransmitter, stuff like that. That should be pretty second nature for you guys. Now, even though neurons are important, we also have glial cells. We spoke about the different glial cells that are both in the central and in the peripheral nervous system. You should know their names and their functions. You know, for example, we spoke about the Schwann cell, right, involved in myelination, as well as the production of neurotrophins for nerve repair in the peripheral nervous system. Speaking of repair, we did speak about how Schwann cells contribute to the repair of PNS neurons. You should know that mechanism as well.
Saw to cut and saw to see in.
Okay, I gotta zap somebody here. Okay. Oops. All right, so I need you guys, a couple people weren't muted. So now it should be muted for everybody. Someone asked a question, is this review recorded? Yes, it is. So it will be recorded and I'll send that out to you. All right. All right, so we spoke about how neurons are repaired, so knowing the different cell types, their functions. Then we started getting into the electrical activity of neurons. And just like many cells, neurons are not any different in the sense that they have a charge difference between the inside and the outside. This is kind of what allows them to be excitable. In a resting neuron, we spoke about kind of the distribution of ions across the membrane. You should know what ions are most abundant outside, which ions are most abundant inside, and how this contributes to our resting membrane potential. We also, of course, that's in a resting state, and of course, part of that ion distribution, remember, is controlled or influenced by those leak channels. Remember I mentioned that we have two main types of channels. We have voltage gated channels, which are activated when a stimulus comes in, but then we have leak channels that are essentially always open that allows ions to leak across. But in order to sort of prevent this continuous leakage, we also have the sodium potassium ATPase pump that brings the ions back. Kind of like if you think of a boat, well, a sinking boat, hopefully you're not in a sinking boat, but if the boat is sinking, you take a bucket, say you're out in a lake somewhere, you take a bucket, fill it up with water and throw it overboard, right? That would be like the sodium potassium ATPase pump, keeping that resting membrane potential so that the ions don't leak across. So membrane potential and ion distribution in response to stimuli, that's what we're talking about, the action potential. You should know the status of the ion channels prior to and during and following a stimulus. For example, when is the voltage-gated sodium channel open? When is it inactive? When is voltage-gated potassium channel open? And when is it inactive? So that's the action potential, kind of like the fundamental response of a neuron, right? It responds electrically, you get this effect. Now, just like with everything else we talk about in our body, there's always factors that influence the function of something, how fast or how slow, kind of regulating it. One of the things we know about action potentials is that how fast the electrical current comes through in the action potential is dependent upon two main factors. Remember, it was the axon diameter as well as the myelination, right? Myelination and as well as diameter. So you should know the factors that contribute to that. I used a really good example when we were talking about squid axons. Remember, they're very large, much too large for them to be in us. But because they're very large diameter, low resistance, they're very fast, but they're also unmyelinated. Whereas in us, we've compensated. We can't have the large unmyelinated axon, but we can have many, many small myelinated ones. All right, so we have this action potential that's going through down the length of the neuron. And of course, at the end of the neuron is a junction between that neuron, kind of a presynaptic neuron, and another neuron, or between that neuron and a muscle, or between that neuron and another glandular cell. This is what we call a synapse. So you should know what makes up the synapse. We spoke about things like, we have synaptic vesicles, which store the neurotransmitter, calcium channels, kind of the role of calcium. That's part of the events at a synapse. Now, what I'm really talking about there are the chemical synapses. The chemical synapses involve the release of a neurotransmitter across the synaptic cleft, right? And of course, we know how it gets across with the aid of these synapsin proteins that promote the translocation of the vesicles to the membrane, and eventually we get exocytosis. And of course, then we know the neurotransmitter goes across to the other side, binds to receptors. Part of those events also, or of course, after the neurotransmitter exerts its effects, it's broken down and recycled. So you should know, again, all the enzymes that are involved with that, with the recycling and the inactivation. Electrical synapses in contrast, we have cells that are communicating with each other through gap junctions, where ions go from a presynaptic cell directly into a postsynaptic cell. All right,